All right, so we're going to get into chapter 6.3 now. So we've already talked about the cell cycle itself, and then more specifically about mitosis, just one simple stage in the cell cycle. Now we're going to get into how does the cell regulate the cell cycle. Basically, how does it make sure that the cell is dividing at the correct time? How does it make sure that it's gone through each of the stages of interphase before it gets to mitosis? How does it go from one stage to the next? So on and so forth. Okay? And then also what can happen when the regulation of the cell cycle kind of breaks down. So cell cycle regulation is necessary for healthy growth. Basically, these are the checks and balances that keep the cell in line, progressing through the cell cycle at a good pace. So if a cell grows too slowly, then the cell will be too small to divide when it's necessary to divide. That might not seem like a huge issue, but this can make you fall behind on growth curves, developmental curves. It can also cause problems with healing, okay? Because cell division is important to heal anything, especially things like cuts, okay? So in order to heal a cut, you have to replace all of the damaged cells that are now gone with new healthy cells. So you have to go through this cell cycle regulation pretty quickly in order to produce more cells to replace what was damaged. So healing is it's obviously really important to your body, and the cell cycle regulation actually helps govern that. If a cell grows too quickly, it'll be forced to divide early. It also means that it will start crowding out other cells. And then you get this massive clump of cells that are all growing too quickly, dividing too quickly, and then they start to lose nutrients because there aren't enough nutrients to go around. And you end up with a lot of issues just simply because the cell grew too quickly. So, like I said, that can... give me a second. Potter's growling. Sorry, Potter heard something and he had to growl at it. But a cell growing too quickly causes issues with obtaining enough nutrients. So then the cells, even though they're healthy, for the most part at least, they start to starve simply because they can't get the nutrients that they need, and maybe there aren't even enough nutrients to go around. So the regulation of the cell cycle is really regulated by internal and external factors. So there are two kind of ways that the cell gets regulated and basically hit with all of these signals. Okay, so this is normal cell growth here. Okay, we'll see abnormal cell growth in a little bit, but external factors include physical and chemical signals. So these can be signals even from other cells, basically saying, hey, I'm right here, you're right against me, there is no room to divide, don't divide. Okay, we're going to have issues if you do, right? So those can be physical signals. You can also get chemical signals coming from other parts of the body, hormones, different types of chemical signals like that. Or you can even get different chemical signals from nearby cells. Okay. You also have growth factors that are proteins that stimulate cell division. So those can be some of those chemical signals that come in and basically tell the cell, okay, you need to divide. You see a lot of these in the healing process. Okay, growth factors will come in and they will have been triggered by the damage and the trauma to the cell tissue with the injury. And then the growth factors will come in and basically tell all of the cells in that area, all right, you need to pick it up, let's get growing, let's get dividing, let's heal this. So most mammal cells form a single layer in a culture dish, which is kind of a problem. Okay, because culture dishes is how we generally grow cells in a lab. But once they touch other cells, they stop dividing. So it's just a single cell, and then they fill up the bottom of the dish, and then that's it. Which, again, that's not how tissues work in the body. Cells actually like to grow in 3D structures. Okay, so they will basically grow on top of each other. We just can't get them to do that in just a simple petri dish. So one of the solutions that a lot of scientists are actually working on perfecting at the moment is these 3D scaffolding structures. Okay, so they have a couple different methods of how they're trying to build these scaffolds. These are just a couple of them, just different ways of actually building the scaffold and then being able to break it down once the cells have grown. But it's still kind of, working on it. But if we can get this to work, there's going to be a lot more things that we could test in a lab and know for sure how they're going to react in that type of tissue in a human body. Okay, because if you can grow them in a way that they act the exact same way that they do in the human body, you're more likely to get an accurate answer out of any question that you ask or test. Okay, whereas with the single flat layer, as that is not how they happen in the body, how they grow in the body at all, it's really difficult to see how tissues would react to different medications, different techniques, things like that. Okay. Another method that some people are working on that they've had some limited success with is to do liquid cultures instead of the traditional agar type cultures. Okay, and the agar is like the gel that we used in that experiment that didn't work so well. But so with these liquid cultures, they are, when they're set up, they're actually put on kind of a turntable sort of thing that shakes them. So they're in constant motion, and the liquid prevents them from forming a single cell layer. So you can actually grow up the cells to a pretty good concentration, and then they act more like a tissue than the single cell layer does, but not so much like the 3D cell layer does, or the 3D cell culture. Okay? All right, so external growth factors can tell the cell that there's enough room around the cell to accommodate the division, okay, which again is important because you don't want to overcrowd the cells, and also that cell division is needed, okay, because even if there's enough room, if the cell division is not needed, or if other cells are dividing and they're going to take up the space or anything like that, then there's no point in continuing to get other cells to grow and divide if all of that's going to get taken care of. Okay, so you've got, not only is there enough room around the cell to accommodate another cell, but also that the cell does actually need to divide. Internal growth factors can tell the cell that the cell is large enough to divide. It's actually getting big enough that it's becoming something of an issue if it doesn't divide. And also that the DNA has been copied, has divided, and it's not damaged. So there are a lot of checks in place with the DNA, specifically because of how important it is that the genetic information is passed on from cell to cell perfectly, or as near to perfect as possible. Okay? So the cell will double check as it goes through copying the DNA that it's copying it correctly and that it's not damaged. Okay? And then it'll also make a double check that, they separate during anaphase. Okay? And then another thing that internal growth factors can tell the cell is that there's actually enough ATP, there's enough energy inside the cell to support the division. Because the worst thing for the cell would be to get halfway through division, run out of ATP, and then they're literally stuck where they are. Can't back out of the division once it starts. And so then they're just kind of sitting around and they die off eventually if they don't have enough ATP. Okay, so two of the most important internal factors are called kinases and cyclins. Okay, they're kind of weird names. They do some different functions inside the cell. So there are 4 major types of cyclins. You have G1, then G1S, S, and M. Obviously, they match up with the phases of interphase. and then into mitosis. Okay. Cyclins actually allow for the transitions between the different stages of the cell cycle. They are those signals that basically say, okay, you have passed this point in the cell cycle, so now the cell is going to start producing that cyclin, and the cyclin is going to tell the cell, all right, well, you passed that point, so let's get ready for the next one, and let's go ahead and go into the next phase. So cyclins are only expressed or produced or created during the stage when they're actually needed to be used. And again, the name of each cyclin basically tells you when they're going to be created and when they're going to be used. So I've got a little graph here. Okay, so the G1 cyclin is actually relatively constant until you get to the M phase. Okay, until you get into mitosis. So the G1 really is what controls growth and normal functions of the cell. Okay, and it only drops off when you get into mitosis because the cell doesn't need to be focusing on normal functions at that point of the cell cycle. Okay, but then you can see it'll pick up right as soon as mitosis is over and it's back in G1. And then when it's nearing the end of G1 phase, that's when you'll start producing G1S. Okay, so that one is specifically to facilitate the transition from G1 into S. So that's the one that kicks off DNA replication. Okay, and then as the S phase is starting to wind down, you get the S cyclin coming in. Okay, that one does peak in G2, so it's a little bit weird, but at this point, you're going to be passing out of the S phase, so you've got DNA replicated, and the S cyclin is responsible for doing those double checks of the DNA. Okay, because the G2 phase is kind of the last resort. If something happened, something went wrong, that's the last place to catch it before you go into mitosis. Okay. Now, with mitosis, you'll notice that the M cyclin starts its production when the S phase starts. Okay, so when DNA starts getting replicated, that's when the cell is already starting to gear up for mitosis, for cell division, because that is the one most important thing that the cell has to go through before it can divide. Okay, and then you'll also notice that the M cyclin peaks right when mitosis starts. So as soon as mitosis starts, its job is done. Mitosis just kind of goes. So you notice that its levels drop off almost instantaneously as soon as mitosis starts. Okay, and then again you go back into G1 and you get the G1 cyclin coming back in. Okay. Now with kinases, kinases are activated by the cyclins. Okay, so this is basically the cyclins carry the message, the kinases act on that message. So the kinases will attach a phosphate group to specific target proteins, and that basically acts as an on switch. So it turns all of these different target proteins either on or off. Okay, so they can make them more active depending on what stage and what cyclin activated the kinase. Okay. Then for those that are kind of, the stage that is ending, it'll make those target proteins less active. So it takes out the ones that are no longer needed. So like at the end of S phase, these kinases will come in and deactivate all of the proteins, target proteins that were used to replicate DNA. Okay, but then they'll also start, kicking up the different proteins that are going to help with cell division. Okay, get those ready. This will cause that reaction within the cell. Now, these kinases don't just target a single protein, they generally target multiple, or they can target other kinases, which will go on and target very specific proteins. And you get what's called a kinase cascade. Okay, so kinase levels actually remain relatively constant during the entire cell cycle, so that whenever that kinase or whenever the cyclin activates the kinase, everything just goes. Okay, so there's no lag time there. But this is where we're going to stop in this video and pick up in the next one.